everyone, my name is Rabia Anwar and we'll be looking for the causes of the death of the fish in the wildlife, and this is associated with the microscopy lab.
I'm Sarah. I'm Kupa.
I'm Richard Ibai.
So the problem was a large number of fish dying in a local pond, and the only environmental change that took place was the excessive usage of salt to prevent ice. Usually there are two types of movements. There are not two types, there are movements in the cell, but two types of them are diffusion and osmosis. Diffusion is when molecules constantly move through a concentration gradient, usually from a lower concentration to a higher concentration. And osmosis diffusion of water across a semi-permeable membrane. If it is water, it will move across the membrane, but if it's something heavier, like charged molecules, they will not move across the membrane. This is a picture of diffusion, and the down one is osmosis. What are the two kinds of situations that occur when the concentration inside of the cell is different than concentration outside of the cell?
Hypotonic, hypertonic.
Can anyone tell what is hypertonic and hypotonic?
Hypotonic is when the cell swells, and hypertonic is when it shrinks.
Yes. So, first one is hypertonic. When the concentration outside of the cell is higher than the inside, the water moves out of the cell to the higher concentration and the cell shrinks. And hypotonic is when the concentration inside of the cell is higher than the outside, the water moves inside of the cell, causing the cell to swell. Both of these situations can be fatal for the cell, and cell cannot survive in these situations. These are the pictures. Low solute concentration, high solute concentration. The A is hypertonic and the B is hypotonic. What is the normal state of a cell? Isotonic is the normal state of cell when the concentration outside and inside of the cell is same. Water molecules move in and out at the same rate. So this is the normal condition for life of a cell. We are doing this experiment because a lot of creatures has exchanged from the earth because of various human activities, and it is very important to know what is bad and what is good for the wildlife. So we want to know what causes the death of the fishes, especially when there is an environmental change by human by putting salt on the roads. So I have a very helpful video that will help you understand osmosis and diffusion. Well, it's just 2 minutes. Can you hear it?
Process allows small molecules such as oxygen and carbon dioxide to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So, yeah. I have a few questions about this. If a cell is placed in an isotonic medium, there will be no net movement of water. Is that true or false? Just watching the video.
False.
Yes, that's right. Swelling of cell is hypertonic or hypotonic? Okay. Yes, that's right. So, based on all this discussion, we hypothesized that the cause of fish dying in the water was the concentration of water changed, which caused the fishes to be hypertonic, and the cells shrank and they died. So I'll go over the materials.
For our experiment, we viewed 4 different Elodia leaves under a microscope, and these are the materials. I won't go too in depth, but we made 4 solutions. We used distilled water and NaCl to make the solutions. Falcon tubes were used to make the solutions. You need latex gloves and lab coat for safety. We use these formulas. I have a question. When you make the solutions, should you add NaCl first or DI water? Yeah.
NaCl.
So we made 0.4 molar, 4 molar, 0.4%, 4% solutions. So 1 gram of NaCl was added to the DI water, actually, was added to the NaCl up to the 25 milliliter mark. We use this 4% solution to make a 0.4% solution, and we did a 1 to 10 dilution, which means basically we used 1 milliliter of 4% and then added 9 milliliters of DI water to it. We basically did the same thing to make the 0.4 molar solution. For 4 molar solution, we used DI water added to the 25 milliliter mark to 5.8 grams of NaCl. So we had 5 different leaves. This 0.9% is the concentration of the solution in the Elodia environment. The other solutions were the ones that we just made. Basically what we did was took a micropipette and we added 1 to 2 drops of solution to each of the leaves. You place the leaves on the glass microscope slide, add a cover slip over the slide, and then wait 5 minutes so the concentration changes inside and outside the cell. And then you view it under 4x, 10x, and 40x magnifications. Does anyone know the proper way to hold a microscope? Yeah.
One hand holding the arm, which is the back of it, and then one under the glass base.
And does anyone know the difference between the coarse adjustment and fine adjustment knobs? Well, okay.
One of them moves the lens and the other one moves the stage.
Yeah. And fine adjustment is used to focus in. So when you're going to do it, first you do a coarse adjustment until you can kind of see the image, and then the fine adjustment knob kind of helps you focus. And you do this with each magnification, and what you see, like if the cell shrinks or swells, you record that into like a data table, or you can draw a picture or something.
And once the experiment was done, we collected the results. When there was water like the solution from the tank added, but it was isotonic and there was no change in shape. And this is what the normal Elodia cell looks like. And like it's important to notice that the chloroplasts are located kind of near the cell wall, but not like exactly right beside it. And this is what it looked like under 400 total magnification. when 0.4 saline solution was added, the cell was hypotonic, so the cell like expanded and water went in, so the chloroplasts went more toward the cell wall, so they were more centered around the cell wall and not inside. And that's, like, this is before it was added and this is after, so you see how they're like collecting toward the walls. When 4%, 0.4 molar and 4 molar saline solution were added, the cell began to shrink, and because of that, the chloroplasts were more inside the cell instead of outside, and more water was leaving out of the cell. And so that's what it looked like, and all the chloroplasts is going inside. And then this was all put into a table, and then you can see that the liquid from the aquarium had no change, so it was isotonic, and then 0.4% swole, and because of that it was hypotonic, and the rest were hypertonic.
Okay, before I conclude, I want to bring everybody back to our point of what's the question. The question was, there was some kind of scenario, and during the winter time, fish die in the pond. And then they figured out that when fish were dying, the roads were being salted. Does anybody know how salted road is going to be? It's going to relate to a pond. Can anybody tell me how salting the road can affect fish in the pond? Yeah.
The runoff, like into a drain, water sources.
Definitely. So that's what happened. So whatever thing my colleagues have been doing, they're just trying to explain the physiology, what's happening in the cells. So what happened was this: the water drained, the salt water drained into the pond, and when it drained to the pond, the water in the pond became concentrated, became hypertonic. So when it became hypertonic, the fish cells, the fish and the wildlife in the pond was more concentrated. The fish and wildlife in the pond lost water. That's the cell. The water from the cells moved from the cells into the pond, and they shrink. And so by that reason, the fish and the wildlife in the pond died. So whatever thing my colleagues have been explaining, we're just trying to tell you that what actually caused the reason why the fish and the wildlife died was because the salt water drained into the pond. When it drained into the pond, the cells of the wildlife and the fish lost water into the pond, they shrink and they die. Any question?